Hi, Anders. Thank you so much for joining this interview. I'm quite pleased to be speaking with you about AGC Biologics. Can you please introduce yourself?
Thank you, Jennifer. Yes, hello. My name is Anders Kai Holm Hansen. I am a principal scientist with AGC Biologics based in Copenhagen. I am part of the customer solution design team who provide technical input at early stages. And when we have incoming projects, we work very hard with our business development colleagues to provide the best possible solution for our customers. And that would entail facility fit, gap assessments, etc.
Oh, yeah. Well, thank you so much. Let's go ahead and get started. So the first question I have is, how do you explain the strategic difference between scale up and scale out to a sponsor that is still in early clinical development?
Thank you, Jennifer. That's an excellent question because at least if you're at early stages of your development, most likely there are some challenges there that you would like to kind of mitigate. I'd say probably demand could still be a bit uncertain at this stage. And you probably also do not really have a feel for, hopefully your project will be a success, but that's kind of part of the whole chain of events to kind of move you down the path towards success and in the end commercial. So to kind of mitigate some of those concerns, the single-use scale-out pathway would kind of be a very efficient way of doing that. So you would, of course, be in a position where you can more efficiently tailor your phase-specific requirements with the effort you put in. Being early phase, you want to invest as little as possible to move towards the next phases where, of course, certainty rises and the outcome of success looks more promising, then you would be willing to invest more. And that is what you can do with a scale-out option.
Brilliant. Okay, so let's talk a bit more about this. So in practical terms, what are the main technical and operational risks that scale-up manufacturing helps to mitigate compared with traditional large-scale stainless steel scale-up?
So when you're looking at you could say the traditional scale-up of going from one vessel size into a much larger vessel size, typically you would impact parameters inside the vessel. That would of course mean that the cell line growing inside the vessel will be impacted by changes to mixing efficiencies, gas and mass transfer rates, and other things that impacts the environment where the cells are growing to generate your product. But if you're using a scale-out version of this, you keep the vessel the same and add more vessels that are duplicating the process, meaning you will not have these changes to environment and making the process more smooth, allowing us to reuse some of the work already done to more efficiently and faster move you to a more, let's say, higher output of your material.
So can you walk us through a recent example where mirrored single-use bioreactors and the six-pack system allowed a program to flex capacity up or down in response to changing demand forecasts?
Yes, thank you, Jennifer. Very recently, we had well, not that recently, but we had a customer which has been with us for some time now, who faced exactly these types of challenges in the beginning. They came to us with a project looking very promising, but very uncertain on the demands and whether or not there would be success. So they started out with a scale of process that is, you would say, on the lower end of the 500-liter process. And as they progressed through the different stages of the project, and we started initiating also, they initiated the trials, clinical trials. Luckily, they saw good results and increases in demand, where we could then very efficiently scale them up to support them through the next round of clinical trials with a 2,000-liter process, and eventually scale them out to support the late stage activities, and are now looking at a multi-pack for them to use for their commercial needs. And actually, we're also now investigating even further to help them with increased capacity by utilizing our network, where we can efficiently also run the commercial process across our sites. We have, in this case, for the mammalian products, we have four sites that could help us out globally. We have one in North America, one in Copenhagen, where I'm based, and we have two in Japan. So here we can now look into providing even better support for the commercial needs down the line.
Brilliant. It's good to hear. So then for complex or atypical proteins, what specific advantages have you seen from combining perfusion at 500 liters with the ability to expand to 2,000 liters in a scale-out paradigm?
So regarding perfusion, so AGC Biologics, we are capable of running both the classic fed batch approach, where you would have a single batch running, or also a perfusion type process, where we would run the process continuously in the upstream. But By combining them, you could imagine a scenario where we would, for typically, you would intensify the seed train, the later stages of the seed train, to allow you to have a very much higher cell density before inoculating the main production stage. And that will allow you to, of course, have a much increased much increased output with a smaller footprint. But you could also turn it around and actually, especially for a little bit the very special molecules, the more atypical ones, or the ones that are not just a standard map, we could also run a pure perfusion process where you would have the benefit of generating much more material even with a fairly low titer process. You would most likely expect that some of these more atypical or complex molecules would not be able to reach the titers that some of the other processes do. But in that case, we could apply a perfusion not just to the we could say N-1 stage, but also as a production stage if needed.
Okay, I got that. So, how does choosing a scale out, single use strategy early in development affect the overall timeline from first in human through commercial launch, especially for accelerated or conditional pathways?
So choosing a single-use scale-out strategy would allow you to move faster, simply because we, as I mentioned before, we reuse a bit the knowledge we generate already in the process. We don't need to do new evaluations of parameters like a complicated scale-up. So we can accelerate the path through, and we can very efficiently scale the output to your needs. So we don't need to generate large quantities of material at a time where there's a bit more risk and uncertainty whether or not the project will progress through the different various stages. So overall, we can streamline the flow of the material, and of course also allow you to better manage your resource expenditures.
Brilliant. So, from a regulatory perspective now, how does maintaining the same bioreactor scale while scaling out influence comparability expectations and the validation PPQ strategy across multiple sites?
So by adopting a scale out, we kind of reduce a bit the validation requirements for the individual equipments as they are similar. So it would allow us to, overall, we can invest a bit more perhaps, but it will in the end give you a much more flexibility to choose a bit the scale you would like to manufacture at eventually. Kind of a balance between how certain are you to reach a specific scale versus how much flexibility would you like. And that is possible with the scale out approach, that we can argue that, of course, instead of having to validate each individual scale, we can do something where we encompass more flexibility on that end. And we have no issues. We have several commercial processes running with scale outs. So it's a very well known process for us, and we see no uh, regulatory issues.
Perfect. That's what you want to hear. And then I'm curious too for long-term implications of early manufacturing choices. Do you see sponsors most often underestimating those implications? And how do you counsel them differently today?
So most often, you could say what we would typically see is that clients may not, and for good reason and understandable reasons, have that long-term view on things. And that is perfectly fine if you're at an early stage of a project. So where we would probably counsel them differently today is that we would try to highlight the benefits and advantages of having the scale out option or strategy that kind of moves the decision making further down the line, so you don't need to tie yourself into a large scale stainless steel process very early. You can delay those decisions and also delay the costs. Similarly, on a more low practical scale, for some of the choices you typically need to do when you are still designing your process, you could end up in a situation where you would be acquiring certain columns with chromatographic resins that would tie you a bit into the process, and you might end up with something that is a little bit too big or too small. So in that sense, we are more able to tailor the processes more specifically to the customer's needs and give them some flexibility around those choices to then later um, have them increase the scale, or have them or give them more output by let's say exchanging chromatographic columns fairly simply, instead of having to go through an entire complex upscaling process in addition to that.
That makes sense. How do your global sites, for example, Chiba, Copenhagen, Yokohama, and Seattle, collaborate to deliver a consistent single use, scale up platform? And what does that mean for tech transfer risk and speed?
So the AGC Biologics network is consistent of the sites you just mentioned. So the main way is that they're of course all aligned with a global quality system. And on a more practical point as well is that we have similar equipment on the lines in Copenhagen and Seattle, allowing us to efficiently transfer that. So in addition to having all the procedures and the quality system aligned, there is regular global alignment meetings trying to give a good overview and ensure best practice in both areas. And we have seen several transfers between sites, whereas especially we had one project where if a customer has an urgent timeline, we may be able to initiate a project at one site, which has free capacity, and then later move it to another site. Or if some geographical constraints show up later in the project, we can also move them quite seamlessly and smoothly between sites because we have this shared infrastructure.
You had talked beforehand about conserving cash and also about for smaller biotechs. So let's say for those smaller or emerging biotechs where they are under pressure to conserve cash, what are the most compelling cost and risk arguments for adopting scale out and single use early rather than delaying that decision?
I think it goes back to the challenge that faces most of us. You want to make sure you are spending your money on the right thing at the right time, and single use and scale out allows you to do just that. So you don't need to engage with a heavy, large scale process until you really need to. So we can flexibly support the entire lifetime of the project in a good way. And especially if you are early, we can make sure you reach those goals and those milestones with as little cash expenditure as possible to move you on to the next phases, hopefully allowing you to continue the ride and cash in with a great success later on.
Yeah, and then so for the final question, looking ahead, how do you see advances in perfusion, automation, and modular single-use facilities further evolving the scale-out model and AGC's ability to launch at clinical scale and scale out post-approval?
That's also a wonderful question. Of course, it's always tricky when you look in your crystal ball. But to me, at least, I think AGC Biologics is already looking ahead. So we are implementing large-scale perfusion processes. So we will be able to manufacture in 2,000-liter perfusion processes. And that allows us to bring molecules to market that previously may have been difficult due to simply not having material availability. Further, it also gives us flexibility to then tailor processes for needs. So we can either have more material out with low titer processes, or we can also take existing processes and simply generate much more material from a much smaller footprint. And then in that way, we would say adapt to the increased complexity that we are seeing. So we are seeing a shift towards more molecules, personalized medicine, an increase in the number of projects. So in that way, we are in a strong position to adapt and support that. And of course, adding also process analytical technologies to streamline the way we manufacture and to allow and to give you, the customers, a much more efficient manufacturing and release at a higher quality is the trends we are looking at. For sure.
Well, thank you so much, Anders, for this interview. Really appreciated speaking with you and hope to talk again soon.
Well, thank you. And thank you for giving me the opportunity to speak on behalf of AGC Biologics on this extremely fascinating and relevant topic.
Absolutely. Thank you again.